What's up, A peeps? Okay, so quick thing, small thing. We're doing proteins today. The cool thing about proteins, between you and me, these are the most important macromolecule. Psst, don't tell the other macromolecules. Okay, so things we are looking for. Don't forget to look about what the structure. What do these things look like? What is the monomer of them? Okay, and there's a lot. What are some examples of the polymers? What are some examples of the monomers? What are the functions, and then what is the directionality. Okay, so let's start this sucker off. First off, proteins. Here's our quick fact sheet. They are macromolecules that are built from any combination of 20 different amino acids. So the monomer unit is an amino acid. There's 20 different ones. You don't have to memorize those for this class, but they are kind of cool, and if you ever take biochem in college, you probably do have to memorize them. And their function, proteins' function, is basically everything. I mean, from making up your bones and your muscles, to catalyzing reactions, to playing a part in your immunity, being part of your blood, hemoglobin. I mean, you name it, proteins probably play a part in it. So we love proteins. They are the bomb diggity, as I always say, and they are a set of different polypeptide chains. Okay, so they have this specific order of amino acids, and depending on the order that we put those amino acids is going to determine the function. And more so even with proteins than any of the other macromolecules, not only do we have to put the amino acids in the right order, but also we have to fold and bend and coil them, because if a protein is not the correct shape, it will not do its correct job. And that's going to be extremely prevalent when we get to enzymes, but for now we'll chill and we'll just talk about the base structure of a protein. Okay, so let's talk about really quick some of the unique features about it. So all polypeptide chains line up carboxyl group on one side, amino group on the other, and the carboxyl group will bind to the amino group, and then it'll link on and it'll create a chain of polypeptide bonds. And so the only thing that identifies the 20 different amino acids is this thing called an R group, or a side chain. Call it an R group when you're writing about it in the AP format. Okay, and so what this R group allows it to do is to fold or bend or coil or pleat. We will see all of these different things, and actually give the protein the 3D shape that is so specific to what its function will be. Okay, so let's look at what that looks like. Here's the amino group. Here is the carboxyl group, and these bad boys line up just in an order: amino, carboxyl, amino, carboxyl, amino, carboxyl. Okay, linked by a carbon. Okay, most of the macromolecules we've seen are carbon-based molecules, all of them actually. And then here we have the R group. Okay, so we might have an alanine bind here, and then it would be an alanine amino acid. We might have a glycine bind here, and then we'll have a glycine amino acid. So this is like a variable. It can be one of 20 different molecular things that bind there and make it unique. Okay, so big thing again, we have the peptide bond, and those things repeat over and over: amino, carboxyl, amino, carboxyl, and that cleans it up. Okay, a single polypeptide chain, however, is not a functioning protein. Okay, we have to do a lot of things to this protein to actually get it to do its job, and what we're doing when we do that is we're making it a 3D structure. Okay, a straight line of molecules can't really do anything. It cannot actually perform a function, however, or do work so often that we see proteins doing. But when we foil them and we create a 3D model, then it has the ability to kind of do some sort of function for us. So again, when we are binding, we see this theme a lot. When we are binding our macromolecules, we go through the process of dehydration synthesis. So that's how we connect that amino to that carboxyl group, is we go through dehydration synthesis, we remove a water, it binds those, and we create a polymer. Okay, So here's the process. We have our amino, our carboxyl, our amino, our carboxyl. These are the R groups. You can see different types of R groups that are binding. And when we're ready to add on another one, we pull out an OH and an H, making that H2O removed, dehydration synthesis. And now we have an amino end and a carboxyl end on a much larger protein, all made up of amino acids. Okay, so look at how they orient because of the way that we've got the amino end and the carboxyl end, and then all the side chains line up together. That's going to be a little bit important as we get to the structures themselves. Okay, so again, sequence of alternating amino groups and carboxyl groups with side chains out to the side. Remember, those side chains can be any of 20 different molecular bindings to make it unique amino acids. Okay, so the protein structure is crucial to its function. Okay, so a functioning protein is not a functioning protein until it is a 3D, uniquely twisted, folded, coiled shape, okay, of polypeptides. All right, so we've got to get it to where we are actually seeing the actual folding, coiling, binding, and so making it a 3D structure before it can actually perform any function. Okay, we've got some organelles that actually help us with that, and we'll talk about those later on in the year. But keep that in mind that like the protein can't just on its own fold the way it's supposed to. That there are cellular organelles that their responsibility is to help with that. Okay, in the more complex things, in the more complex organisms. Okay, so proteins will account for about 50% of the dry mass of most of your cells. Okay, so they are huge for the macromolecules. Okay, they make up your muscles, they make up your bones. Okay, they have enzymatic properties, as in they can help catalyze chemical reactions. They are support proteins. You see those in your bones. You see that in like silk of spiders. You can see that in different horns and hair and other little appendages and things like that. It's your connective tissue. You can see them as storage units. Okay, like for instance, there are proteins in milk, and that helps us store energy and things like that. There's proteins in plant seeds that keep them alive even though they are dormant. Okay, there are transport proteins which help us move substances. So like we'll see those in cell membranes, but then there's also unique ones like hemoglobin which move oxygen all throughout the body. There's hormonal proteins. Now we've seen hormones before as lipids, but now we've also got some in the form of proteins. We've got receptor proteins, which are responsible for like signaling. Okay, they're going to take in some information and then they're going to make something within the cell actually happen. We've got contractile or motor proteins. These are your muscles. These are like the flagella on cells to help them swim, or cilia on cells to help them move around. We've got also like there's things like contractile vacuoles, which helps us eject water in some certain cells. And then obviously we have some that are in our immune system, like we have antibodies, and their job is to help us fight off viruses and bacteria. And so we have a lot of different functions of proteins, a bunch. Do you have to know every single one right this second? No. Are we going to talk about all of these? Yeah, at some point throughout the year. So please just like have that idea in your head that protein's probably the most important macromolecule as far as your test is concerned. Okay, so let's talk about how we go from a chain of amino acids to an actual functioning protein to actually get all of those functions we just talked about. Okay, so we go through 4 stages or 4 structures of a protein before we can even get to the 3D coiled shape that we know and love as a functioning protein. Okay, and it's pretty simple to kind of generate. It's a primary structure, a secondary structure, a big surprise, tertiary structure, and then finally when it's a functioning protein, it's a quaternary structure. Okay, here's that same thing, a little bit different. You can see that there's a primary structure, okay, a more complex secondary structure, a tertiary structure, and then the quaternary structure. All right, so how does it actually go through all of those steps? Okay, well, the primary structure is the easiest because it's just a chain of amino acids. So that amino, carboxyl, amino, carboxyl, amino, carboxyl. And remember, each protein is going to have a unique sequence of those 20 amino acids. Now, where do we get that sequence code from? Our DNA. Okay, so we'll talk about that a little bit later. But just for the sake of the proteins, I like to mention it just so that you're aware of how we got that unique pattern of amino acids. Okay, it came straight from the DNA of whatever the cell is. Okay, so the primary structure is just your line of amino acids all lined up. Okay, amino acids being the monomer of a protein. Okay. Now, the secondary structure, we start to get a little bit more complex, okay? So, we went from a line of amino acids, and I like to do a little dance, so I've got my little line of amino acids, and then in the secondary structure, we're going to do one of two things, okay? Or both things. We will have a coiling of a alpha helix, okay, where it will fold on top of itself. Okay, so we'll see them kind of like start to twist and form and go like this, and those R groups are what are really helping us kind of fold on top of ourselves and bend and coil. Okay, so we'll create alpha helixes. Okay, the second thing we might do is instead of being a straight line, we might kink. Okay, and so this is actually a beta pleated sheet. Okay, so these are the two things that are actually happening in our secondary structure. Is our straight line of amino acids is folding alpha helix or kinking beta pleated sheet. Okay, this is all the result of different hydrogen bonds that are in between our polypeptide backbone. Okay, so now once we've got our hydrogen bonds and interactions between R groups, we can move to a tertiary structure where we are either further coiling. Okay, so we were doing this number right here in the beginning. We were bending, we were coiling. Now we're really going to get those R groups involved, and we might actually fold in on ourselves like this. Okay, so now we are a little bit more 3D, but we're still just that one line of amino acids. And this is through covalent bonding called a disulfide bridge. Okay, so a disulfide bridge is actually going to give a singular link of amino acids, one amino acid chain, a 3D shape with a disulfide bridge. Okay, so here we go. We can see our different bondings. Here we might have had like this is where we were having hydrophobic interactions and van der Waals interactions. That's kind of funky and crazy. And then we're going to fold all the way on top of ourselves like this and create disulfide bridges to create, again, a 3D structure of just a singular amino acid chain. Okay. And last but not least, once we get that tertiary structure, we're really happy about it. But now what we're going to do is we're going to take a second amino acid chain who is also in its tertiary form, and we're going to stick those two things together to get something called a quaternary structure. And now, if it's in the right shape, we have a functioning protein. Okay, so recap all from the very beginning. We had a amino acid chain of stuff. We alpha helixed and beta pleated to create a secondary structure. We then disulfide bridged through hydrophobic reactions. These are called van der Waals reactions. And that actually gave us a tertiary structure, which was 3D, but only one chain of amino acids. And now finally, we put two of those together, and we have a quaternary structure, which is a polypeptide chain, folded, coiled, twisted, and binded to a second tertiary structure, quaternary structure. Okay, so that is huge for us, because if that protein does not bend and fold and get to a quaternary structure that is meant to be the way it's meant to be, okay, if it does not develop in its correct form, it will not function properly. Okay, so a great example we like to talk about is our immune system, because it's clearly a form fits a function. Okay, so in our immune system, we have these proteins that bend and fold, and they're called antibodies, and their job is to recognize these nasty little booger brains called antigens. Okay, sometimes you'll hear this as a pathogen, and pathogens can be antigens, and antigens don't have to be pathogens. I know it's crazy, but these are just like foreign substances that your body doesn't want in it. And so an antigen might that is recognized by an antibody, this antibody can call in backup and destroy these antigens. But we have to recognize them first. So if this doesn't fold and bend the way it's supposed to, these particular foreign substances don't get recognized, they're going to mess us up, okay? But if our protein did its job and it's able to recognize that antigen and it bended and folded the correct way to recognize that antigen, then it's just like a lock and key. We recognize the antigen, we call in for backup, and then we destroy those little foreign agents. Okay, some other examples that we like to talk about. So we've got a polypeptide chain here. We can see that they bend and fold this way. We're going to get a really unique shape to create collagen. We can also see in hemoglobin, we can get a very unique shape so that it is performing the correct job that it's supposed to. And in it, you can see alpha helixes, you can see beta pleated sheets, you can see that there was a singular amino acid chain that bended and folded the way that it was supposed to, and it got to a tertiary shape, and then those tertiary shapes combined to create a quaternary one that was doing something, an actual functioning. Okay, here's some videos. It just goes through kind of like the progression of making those. Not necessary that you watch them, but if you want a little bit more help with it. And so it really does depend on how we bend and fold. So collagen, for instance, it's really fibrous. It's kind of like a rope there for us, which we definitely will need in our bodies. You'll kind of find it in your body, like helping out muscles and things like that. Versus hemoglobin, which it had more of a bowl shape, right? Because its job is to carry substances, and so it's going to be a little bit different, and it's going to perform a different function. Okay, so form fits function. Again, we see that. So an example of form not fitting function, we have a problem here. There is a disorder. It's called sickle cell disease. Okay, now sickle cell disease is caused by a mutation, and remember we said the code of amino acids is determined by the line of DNA. Okay, of our DNA, that template. Okay, so anytime we have any slight changes to that code, we might insert the wrong amino acid. We're going to develop the wrong shape. We're not going to perform the right function, okay? So here you can see a correct chain of amino acids determined by that code. We go through all of our structures. In the quaternary structure, it is this nice little bowl so that it can carry oxygen for us. Versus here, okay, and I need to move my head, versus here, you can see that we have a little mutation right here. The code was incorrect, so the amino acid sequence is wrong. Therefore, we have a binding in our quaternary structure that is wrong. Therefore, we don't have that pretty shape that we have. So this bad boy is not going to be able to carry blood the way that it should. So it is not going to perform the function that it needs. Now, luckily, people who are carriers of sickle cell, they have some of these really pretty ones that can still carry oxygen for them. So they'll be okay. But these guys that are in them won't. So they're going to be way less efficient in their ability to carry blood. Okay. So now when we talk about proteins, we do care about their shape. Okay. So there are some problems that can occur with proteins, such as denaturing, which means that it's just going to lose its shape, and therefore it's not going to be able to perform its function correctly. Denature just means change the shape of the protein. Okay, now this can be caused by a lot of different factors that are environmental factors for our protein, such as the pH has changed, or the temperature has changed, or the salt concentration is not where it wants to be. Okay, and it'll just cause that protein to unravel. Some of the bonds will break. It won't have the right shape. Therefore, it's not going to be able to perform the correct function. Okay, so again, that's called denaturing. We're going to see that a lot. Okay, so it comes up for sure. Okay, all right, here's another example. Now, we have denaturing. Sorry about the bell. we have denaturing occurring, okay? Here is our normal protein. It's coiled, it's folded, it's doing exactly what it's supposed to. It's 3D. Now, once we denature it, again, those bonds break apart. It becomes this ribbon. This can't actually perform the function that it's supposed to do, okay? My video head keeps getting in the way, okay? So now we have a denatured protein, which is not doing what it's supposed to. But if the environment were to return to the normal conditions, those bonds might reform, okay? It might not be the exact shape it's supposed to be, but it'll be pretty dang close, and it'll be able to become a normal protein again. So there is a process of denaturing, but we can also, if we return the protein to where it's supposed to be, we can often renature it sometimes as well. Okay, so next video we're going to do enzymes, which are still proteins, but they are way more in depth. So I just wanted to give you a heads up for that. I am hoping you guys have a great rest of your day, and I will see you later.